The cutting of DNA fragments by using restriction endonucleases results in producing fragments of DNA. These fragments of DNA can be separated by a process called as gel electrophoresis. In this gel electrophoresis, these fragments are separated based on their sizes. Now, this separation technique is based on the movement of charged particles when electric field is applied, and here the movement of these particles occurs through gel media. It's called gel electrophoresis. So here the gel that we are using in this technique is agarose gel. Agarose gel is extracted from seaweeds. Fine. If we have DNA fragments which are separated by using which are fragmented by using restriction enzymes, and through gel electrophoresis we can separate these DNA fragments, right? So the basic requirement for this is the gel plate and a power supply. So let us talk about the few basic things of this technique. So here we are going to load the DNA fragments to a gel plate, and this gel plate is placed in a casting tray. So this is casting tray. In this casting tray we are going to place the gel plate. Next, in the gel plate, one end of the gel plate is with negative terminal, the other one is with positive terminal. Other is positive terminal. So we are going to load our DNA fragments at the negative terminal. Reason behind this is DNA fragments are negatively charged. So as a result, we are going to place them on the negative terminal, right? For loading our DNA, we are going to form wells. So we are going to form wells for loading DNA. So next question is, where do we form wells? Wells are created, so you can just visualize the wells. Wells are created near the negative terminal because we are going to load the DNA fragments at the negative terminal, because DNA molecule is negatively charged because of the phosphate group present in it. Fine. After loading the DNA fragments on the gel plate, then the power is turned on. Then the fragments will start moving through this electric field. Larger the size of the fragments, lesser or slow the movement is. Smaller fragments move faster. This is how all the DNA fragments are separated. So the size of the DNA fragment is smaller, it moves faster. Size of the DNA fragment is large, it moves slowly. So once the DNA fragments are separated, then the power is switched off. Switch off and switch on. Power is switched off. Then all these DNA fragments are separated on the gel plate. Now, so these DNA fragments which are separated on the gel plate are compared with standard DNA chart, which is known as DNA ladder. This is DNA ladder, which is a standard chart where all these fragment lengths are present. So comparing the DNA fragments on the gel plate with the standard fragment length, we can know the length of our DNA fragment, or we can find out our desired gene based on the length. Fine. These DNA fragments now on the gel plate cannot be seen. So as a result, these DNA fragments are treated with a compound called ethidium bromide. Exposing to UV light, we can see DNA fragments as bright orange colored. Bright orange colored. When once, based on the DNA ladder, the standard length, if we can identify our desired gene, this is manually cut from the gel plate and is extracted. This technique is known as elution. So this is how, by using a simple technique of gel electrophoresis, we can separate our DNA fragments and our desired gene can be extracted from it. So this technique is based on the size of the DNA fragments. This is also called as density gradient method of electrophoresis.